Hello students, welcome to EPG Pathshala. I am Farhan Jalis Ahmed from Hamdard University, New Delhi, and today we are going to discuss on the module materials which are used for colloidal drug delivery systems under the paper Novel Drug Delivery Systems Part 2. The objectives of this module is to understand about the classification of various materials which are used in the preparation or development of colloidal drug delivery systems, to explore the ideal properties of polymers which make them suitable carrier for the drug delivery systems, to discuss the advantages and limitations of natural materials over synthetic materials, to study the physical, chemical properties of individual polymeric material. Now, colloidal drug delivery systems, they are vesicles which are forming and having a particle size ranging from nanometer to micro size range. These carriers can increase the surface area for dissolution and thereby improve the bioavailability of the poorly water soluble drugs. They are widely employed in the drug delivery systems to protect inactive constituents and playing a key role in the delivery of therapeutic agents. Various natural and synthetic materials are widely used in the formation of these systems are. Now, biomaterials are generally substances which are other than food or drugs contained in the therapeutic or diagnostic systems that are contact with tissue or biological fluids. No longer is the treatment of diabetes, asthma, cardiac problems, cancer and other diseases based only on conventional pharmaceutical formulation. What are conventional formulations? Like tablets, capsules. Now we are using specialized systems. Biomaterials already contribute greatly to the improvement of better healthcare system. Now, polymers have played a very important integral role in the advancement of drug delivery technology by providing a controlled release of therapeutic agents in a constant doses over a long period of time. It can reduce the dosage and it can also control the release of both hydrophilic drugs as well as hydrophobic drugs. If you look at the ideal properties of a polymer which are required to be formulated as a drug delivery system, the most important property that these polymers or materials has to be biocompatible. If they are not biocompatible, they can interfere or they can interact with the body fluids and they can cause various types of reactions. Next important property is they have to be biodegradable. So they can be easily excreted from the body and consumed by the body and should not stay inside the body forever. Next most important aspect is their safety. They have to be safe, they have to be non-toxic, and they should not cause any type of allergic reaction or any type of toxicity inside the body. Other important property is their aqueous solubility. They have to be soluble in aqueous media, so can be easily formed into solution and can easily release the drug inside the body by getting solubilized in body fluids. Then they have to be chemically inert, so they should not interact with the drug molecules. They should not interact with other systems or other excipients in the formulation. Then their easy availability and low cost. Low cost is also very important, so we can control the overall cost of the product. Then they should be non-immunogenic. They should not cause any immunogenic reaction inside the body when they are injected parenterally or into the muscular system. Then they should be open for structural modifications. So by modifying their structure, we can change their properties. We can change the number of molecules which can be, or different types of molecules, different types of drugs which can be incorporated in these polymers. Now let's talk about microparticles and nanoparticles. The utilization of micro and nanoparticles in the drug delivery system is a strategy that basically means to optimize the therapeutic effects while also minimizing the adverse effects. Most of the polymeric materials which are used for the preparation of particulate carriers, they have to be and they are biocompatible and biodegradable. If you look at different types of polymers which can be used for microparticles or nanoparticles, they are classified into two classes: natural polymers and synthetic polymers. Natural polymers, which are most commonly used and used by the scientists and also used by the industry, the first is chitosan, gelatin, alginate, albumin and pectin. There are other natural polymers which can be used, but these are the most commonly used. Then if you look at the synthetic polymers, we have polylactic coglycolic acid, polylactic acid, poly-E caprolactone. They are most commonly used and they are considered as safe polymers for delivery or for formulating into microparticles or nanoparticles. Chitosan is one of the most accepted biomaterial and it is generally recognized as safe polymeric material in the area of colloidal drug delivery system. Chitosan is basically a deacetylated derivative of chitin which is present mainly in the shells of molluscs and crustaceans, fungi cell walls and cuticle of insects. Its backbone contains an amino group that builds up a positive charge on the surface of the chitosan and have cross-linking ability with number of cross-linking agents like tripolyphosphate, sulfate and citrate. Chitosan is potential carrier for colloidal delivery systems such as microspheres, nanoparticles, hydrogels, films and beads. Now coming to gelatin, gelatin is a very old material which is I think in use since ages as a capsule. Gelatin is a naturally derived protein which is obtained by the hydrolysis of collagen. US FDA has approved gelatin as a GRAS polymer. Gelatin is soluble in hot water, glycerine and dilute acidic solutions. It is biodegradable, non-toxic, biocompatible, non-antigenic and it has high nutritional value, it has low cost and it has very good cross-linking ability which makes it suitable for the colloidal carrier system. Gelatin has an enormous potential to be used for the preparation of drug delivery systems like microspheres, nanoparticles, hydrogels and liposomes. Now talking about alginate, alginate is also a very popular material. It is isolated from a variety of algae, mainly brown algae such as macrocystic pyrifera, Laminaria hyperbora and Ascophyllum nodosum. And it is also classified as a GRAS material by US FDA. Alginate is soluble in water. It forms a viscous colloidal solution. It is extensively used colloidal material because of its biocompatibility, biodegradability, sol gel transition property, non-toxicity, easy availability and chemical versatility. And it is widely utilized for the preparation of microparticles, nanoparticles and hydrogels. Now other biomaterial which is used is albumin. Albumin is a soluble protein of the circulating system and it is involved in the maintenance of osmotic pressure, binding and transport of nutrients to the cells. Albumin is freely soluble in water, diluted solution and makes it an attractive molecular carrier for the encapsulation of a wide range of drugs and serves as a depot and transport protein. It is effective colloidal carrier for the delivery of therapeutic agents in the form of nanoparticles and microparticles. Pectin is another biomaterial which is water soluble and it is a linear polysaccharide. It is obtained as a component of plant cell wall. Pectin with low degree of methylation forms gel in the presence of multivalent ions, whereas pectins with a high degree of methylation forms gel in the acidic media. Pectin is a non-toxic, biocompatible colloidal carrier for the encapsulation of the large number of drugs. The pectin-based hydrogels, films, microparticles and nanoparticles, they has been used for the targeting of drugs to various organs, various parts of the body. Another popular biodegradable material is polylactic coglycolic acid, which is known as PLGA. It is a copolymer of polylactic acid and polyglycolic acid and it is soluble in chloroform, tetrahydrofuran, acetone and ethyl acetate. US FDA has approved PLGA as a GRAS material and it is one of the most effective colloidal polymer for the controlled delivery of drugs. PLGA is hydrolyzed in the body and breaks down into biodegradable metabolites, lactic acid and glycolic acid and metabolized in the body via the Krebs cycle and removed as carbon dioxide and water. Now polylactic acid is a synthetic polymeric material which is having a GRAS status. It can be employed in the manufacturing of colloidal carrier based nanoparticles and microparticles. It is utilized for the encapsulation of a variety of therapeutic agents as a rate controlling polymer due to its biocompatibility, biodegradability and absence of any major toxicity. Polylactic acid is breakdown into monomers of lactic acid and body fluids and lactic acid and it is a natural byproduct of anaerobic respiration. Further, lactic acid is converted into glucose by the liver and glucose is then used as a source of energy in the body. Now talking about polycaprolactone, it is a biocompatible, non-toxic, biodegradable polyester which is used for the preparation of nano and microparticulate systems. It is soluble in chloroform, dichloromethane and benzene, and it is gaining interest as a choice of material in the drug delivery systems and long-term implantable devices or depot systems. Dendrimers are nano-sized colloidal systems which can be prepared from multiple branched monomers, which are amidoamines, and is the well-known dendrimer used for the synthesis of dendrimers known as starburst dendrimers. Dendrimers synthesized from poly or polypropylene amine usually have polyalkyl amine as end groups, and numerous tertiary tris-propylene amines present in the interior portion of the dendrimer. Polyester and hyper-branched polymers have been found to be non-toxic and are extensively used in the formation of dendrimers. Hyper-branched polymers belong to a specific category of synthetic tree-like macromolecular structure which contains dense branch structure with a very high branching density and having potential of branching in each repeating unit. Hyperbranched polyglycerols and hyperbranched polymers containing poly or oleoethylene glycols and other biodegradable materials are potential candidates for colloidal drug delivery. A very interesting nanosystem is nanodiamonds. They are carbon nanomaterials which are condensed octahedral diamond-like structures which are in the size range of about 1 nanometer to 150 nanometers. Nanodiamond is a allotropic form of carbon. Among all carbon allotropes, diamond can be considered as the most suitable colloidal material for nanodiamonds and graphite, and it is one of the most stable and common allotropes of carbon. Liposomes are flexible and versatile colloidal carrier systems which resemble with the lipid membrane of the living tissue and can be prepared from the natural or synthetic phospholipids or polymers which are biocompatible and biodegradable. Now let's discuss the composition of liposomes. Liposomes are comprising of phospholipids and cholesterol as the main two materials. Phospholipids are basically most important composition of the biological membranes and highly accepted material for the liposome preparation. Lipid content consists of phosphorus group known as phospholipids and utilized in the manufacturing of different types of formulations like emulsions, mixed micelles, and liposomes. Different types of lipids and lipid mixtures can be used to obtain a certain type of liposome. Phospholipids are surface active, amphiphilic material consisting of a hydrophilic head and a lipophilic tail and are used for emulsification as wetting agent, solubilizer, and base material for liposomes. The phospholipid molecule comprises a glycerol backbone which is esterified in the position 1 and 2 with fatty acids and position 3 with phosphate. The amphiphilic nature and the insolubility in water and solubility in organic solvents makes phospholipids as ideal candidates for the formation of biological membranes. The phospholipids can be divided into different types of phospholipids, natural or synthetic. Now cholesterol. Cholesterol is basically a sterol and its derivatives are frequently utilized for many reasons like decrease in fluidity of the lipid bilayer membrane, decrease in membrane permeability towards water-soluble molecules, and stabilization of plasma membrane. Lipid-based formulations without addition of cholesterol undergo physical interaction with plasma proteins. This interaction causes extraction of bulk lipid material from the liposome's vesicles, therefore breaking the outer layer of the vesicles, which causes physical instability of the formulation. That is why cholesterols are used to further strengthen or stabilize the liposomes. Now phospholipids are mainly of two types, natural phospholipids and synthetic phospholipids. Now in natural phospholipids, they are naturally derived lipids at very large scale from natural plant animal sources such as soybean, sunflower seed, rapeseed, flaxseed, egg yolk, and milk. The sources for natural phospholipids are egg yolks and soya beans, which consist of phosphatidylcholine, phosphatidylethanolamine, phosphatidylinositol, and sphingomycin. For large-scale production of soya lecithin and crude soybean lecithin act as a starting material containing high phosphatidylcholine content. Liposomes stability is an important parameter for the selection of lipids composition in liposomes preparation and affected by type of lipid used. Phospholipid isolated from natural sources generally have low cost as compared to lipids which are derived from synthetic or semi-synthetic source. Although phospholipids from natural sources are very popular because of their safety issues. Now, synthetic phospholipids are derived from natural lipid sources by chemical modification in their polar and non-polar groups. This gives different well-defined phospholipids. Synthetic phospholipids are dipalmitoyl phosphatidylcholine, diesteroyl phosphatidylcholine, dimyristoyl phosphatidylcholine, dipalmitoyl phosphatidylethynolamine, dipalmitoyl phosphatidylserine, dipalmitoyl phosphatidylglycerol, and dioleuryl phosphatidylcholine, etc. Nuosomes are non-ionic surfactant-based vesicles forming biliary structures and which can be used for the encapsulation of a variety of hydrophilic and lipophilic drugs. Nuosomes, nanoliposomes or pharmacosomes. Nuosomes are basically can be defined as nanoliposomes also and pharmacosomes also. And all the three, they are mainly use two main materials. One is non-ionic surfactant, other is cholesterol. So nuosomes consist of mainly two components, non-ionic surfactants and cholesterol. Various physiochemical properties of nuosomal vesicles like size of the vesicle, lamellar structure, shape, stability in the body fluids, and controlled delivery of drug at the target site are affected by their composition, which is used for their preparation. So the amount of cholesterol being used, amount and type of non-ionic surfactant which is being used, it will affect the stability as well as the release profile of all these three formulations. Non-ionic surfactants consist of hydrophilic head and hydrophobic tail, which form a closed biliary vesicles in the aqueous media. You can see in the figure, they also have low toxicity, less hemolytic property, and less irritating to the cellular surfaces. These act as solubilizers, emulsifiers, and permeability enhancers. Now let's discuss various examples of non-ionic surfactants. Surfactants, they can be of three classes: ether and ester linked surfactants, sorbitol fatty esters, and polysorbates. Ether and ester linked surfactants are polyoxyethylene alkyl esters having hydrophilic and hydrophobic groups, which are attached with ether linkage. Commonly used ether linked non-ionic surfactants for nuosomes preparations are polyoxyethylene 4-lauryl ether, which is known as bridge 30, polyoxyethylene cetyl ethers, which is known as bridge 58, and polyoxyethylene stearyl ethers, which is known as bridge 72 and 76. These surfactants are also used in the formulation of solid lipid nanoparticles. Next class of non-ionic surfactant is sorbitol fatty esters. These are the derivatives of polyoxyethylene esters to solubilize essential oils in water-based products. Ester of plain, which is without pegylation, sorbitol with fatty acids are known as SPANs. SPAN 20, 40, 60, 80 and 85 are examples of sorbitol esters, which are mixtures of partial esters of sorbitol and its mono and dianhydrides with oleic acids. Nuosome prepared by higher molecular weight SPANs are highly stable and shows less leakage, so content remains safe inside these systems. The third substance is polysorbates. Polysorbates are twins and are liquid derived from pegylated sorbitol acidified with fatty acids. Twin 20, twin 40 and twin 60 are the most commonly used twins in the nuosomal preparations. Cholesterol provide rigidity and stability to the vesicle structure. It is used in the preparation of liposome. It is incorporated in nuosome formulations. Cholesterol is a natural steroidal component mainly used in the high molar ratio with non-ionic surfactants as membrane stabilizer of the vesicle. Micelles are macromolecular range type of system which is designed from amphiphilic synthetic or graft copolymer consisting of a spherical hydrophobic inner core, and it has an outer shell. Polymeric micelles are formed in aqueous solution from block or graft copolymers. Copolymers which are used in the preparation of micelles may be diblock or triblock depending on the requirement of the drug delivery system. Diblock copolymers represents XY type, where X represents the hydrophilic portion and Y represents the hydrophobic portion. On the other hand, triblock copolymers contain two types of polymers XYX and three types of polymers XYZ. Main purpose of formulating polymeric micelle is to entrap the drug physically or covalently to the hydrophobic portion of the micelle. This linkage causes increase in solubility of poorly water-soluble drugs and protection of several unstable drugs from chemically and metabolic degradation. Selection of material for the preparation of polymycicle is an important factor which directly affects the characterization parameters of the micelles like drug entrapment within the micelle, drug loading, and in vitro drug release profile. Fluoronics polyesters such as PLA, copolymers of lactic acid and glycolic acid, hydrophobic polyamino acids, and PCl are recognized as the core forming blocks of polymeric micelles. PG is the most suitable hydrophilic polymer used in the development of polymeric micelles, and polyanvinyl 2-pyrrolidone and polyacrylic acid are used as PG alternatives. Polyethylene oxide or polypropylene oxide copolymer, which is popularly known as PEO and PPO, it is an amphiphilic triblock copolymer which is comprising of hydrophilic ethylene oxide and lipophilic propylene oxides. These products are marketed as plurononic, saniprononic, and poloxamine in the market. These triblock copolymers have significant aqueous solubility, biocompatibility, biodegradability with minimum or no toxicity, and they have greater capability to develop polymeric micelles, macro or microse emulsions, and other liquid crystalline phases which can be used for the drug delivery systems. Polyethylene glycol is a FDA-approved hydrophilic non-toxic polymer for the preparation of various colloidal formulations. Polyethylene glycol reduces opsonizing action of the macrophages, and thereby it increases the probability of active molecule to target the desired site inside the body and improves the pharmacodynamic behavior of the drug. PEG is most accepted material in the preparation of micelles, surface modification of the nanoparticles, and it is mainly due to its diverse physiochemical characteristics. If we talk about polyesters, among different colloidal polymers like polypeptides and polysaccharides, the bilateral polyesters are used for the polymeric micellar formation. A variety of hydrophobic polyesters can be used for micelles development. They are including PLGA, PCl, and PLA. Degradation rates of these materials vary according to their chemical composition, and it can be used for drug targeting and property. These are FDA approved materials with diverse biomedical applications for the human healthcare system. Polyanilyl 2-pyrrolidone, PVP is a water soluble polymer. It is biocompatible and biodegradable polymer which is used in the preparation of colloidal drug delivery systems. PVP can increase the circulation time of peptides, proteins, and is a good solubility enhancer for various poorly water soluble drugs because of its complex formation. It can form the complex with drugs and make them soluble. PVP is a self-assembled lipoprotectant, and it is used for the incorporation of number of drugs in the inner core and the outer shells with a high loading capacity. Now if you look at the figure of microemulsions and nanoemulsions, microemulsion or nanoemulsion, they are mainly comprising of surfactant, penetrating agent, and solubilizer. Microemulsions are stable isotropic liquid mixtures of oil, aqueous phase, and surfactant. However, sometimes a co-surfactant is also used for the preparation of microemulsion. Variety of oil phase and surfactants are utilized for the formulation of microemulsion systems. All these materials, they should be biocompatible and non-toxic in the nature, and there should be a balance, and their values should be properly calculated and optimized. All microemulsions and nanoemulsions, they are comprising of three main components: oil phase, surfactants, and co-surfactants. Oil phase is basically saturated fatty acids of lauric acid, capric acid, myristic acid, and unsaturated fatty acids of oleic acid and linoleic acid. Ethyl or methyl esters of lauric, myristic, and oleic acids are commonly used as oil phase for the preparation of microemulsion. Oil phase in emulsion system provides ability to penetrate and improves swelling of hydrophobic portion of surfactant layer. Selection of suitable oil phase for emulsion formation depends upon the drug solubility. So depending upon the drug solubility, these surfactants and co-surfactants and oil phase are selected. Next component is surfactants. The surfactants are amphiphiles consisting of polar head group and hydrophobic tail, which lowers the surface tension between two phases, aqueous phase and oily phase, and favors the preparation of microemulsions. Surfactants with low HLB value helps in the preparation of water and oil type emulsions, while surfactants with high HLB value are used for oil in water type emulsion. Polysorbate 20, 80, Ispan 80, polyoxol 40 stearate, and various polyglycolized glycerides are well-known surfactants which are being utilized for the formulation of microemulsion. Another important component of this system is a co-surfactant. In most of the cases, microemulsion system, single chain surfactants are not be able to reduce the interfacial tension between oil phase and aqueous phase. Therefore, co-surfactants such as propylene glycol, butanol, pentanol, and hexanol is used to produce the transparent emulsion system. By using these co-surfactants, we are getting a more flexible system where we can modify the HLB value according to the type of drug or type of oil which we are using. Now let's summarize whatever we have covered in this presentation. Chitosan is suitable for effective encapsulation of both macromolecules and low molecular weight drugs. US FDA has approved gelatin as a GRAS polymer, therefore it has an enormous potential to be used for preparation of drug delivery systems like microspheres, nanoparticles, hydrogels, and liposomes. Alginates are extensively used colloidal biomaterial owing to the biocompatibility, biodegradability, sol gel transition property, non-toxicity, easy availability, and chemical versatility. Polylactic glycolic acid or PLGA is one of the most widely used synthetic polymers which have been used to formulate protein and peptide based nanomedicines, vaccines, and also for gene delivery. Polylactic acid is another synthetic polymer material having GRAS status and can be employed of nanoparticles and microparticles to achieve controlled release behavior. PCL has gaining interest as a choice of material in the drug delivery system and long-term implantable devices. Fluoronics, polyesters, polyamino acids, and are recognized as the core forming blocks of polymeric micelles. Polyethylene glycol is the most suitable hydrophilic polymer used in the development of polymeric micelles and reduce opsonization. Opsonization happens when these systems are injected into the parenteral system. When they are injected in the parenteral system, these microemulsions or nanoemulsions, they are opsonized and they are cleared out by reticuloendothelial system. It is well known that some water oil surfactant systems can be optimized to give three phases. The middle phase is a thermodynamically stable microemulsion. Three-phase system can also form when most of the water is replaced by polyethylene glycol. The phase behavior of such PEG-based systems with undecyl and olealyl ethoxylates as surfactant has been studied by researchers. The PEG reduces the temperature sensitivity and shifts the optimum to higher EO numbers, that is, higher HLB values. In single-phase oil-in-water microemulsion, the polyethylene glycol also reduces the temperature sensitivity, though the effect seems to be less than for three-phase system. It also promotes the solubilization of higher molecular weight oils such as triglycerides. Saturated fatty acids of lauric acid, capric acids, myristic acids, and unsaturated fatty acids of oleic acid and linoleic acid, ethyl or methyl esters of lauric, myristic, and oleic acids are commonly used as oil phases for the preparation of microemulsion or nanoemulsion. So the students, let's conclude what we have understood from this module. We have learned about natural and synthetic polymers, phospholipids, surface active agents, polyesters, copolymers, etc. And we have learned that they have been used as excellent backbone materials for colloidal and supramolecular drug delivery systems. The biocompatibility and the biodegradability, low toxicity, and increased stabilities are the ideal properties of a promising carrier for colloidal drug delivery systems because it has to be biocompatible and as well as biodegradable. In future, the modification of in structures and modification in physiochemical characteristics of colloidal materials will improve the drug loading, and it will also improve the its release. The colloidal carrier systems will be a promising approach as next generation drug delivery systems, and in future most of the biotechnology based drugs will be formulated as colloidal drug delivery system.